in the UK with his PhD from Cambridge University, came to the United States. In 1993, he joined Advanced Tissue Sciences, and when that got folded in Smith and Nephew, he became a Smith and Nephew employee. I guess that's one way to
put it.
So, but he's been with the pioneering efforts in the U.S. in terms of developing skin substitutes and in the context of commercializing cell-based product. And this morning he's going to talk about development of a skin substitute. So, Jonathan, it's all yours.
Thank you very much, Bob. So I'd just like to begin by thanking Bob and Myrna for inviting me to give this talk and for bringing me to such a wonderful place. This talk is going to be something of an introduction to a biotech view of tissue engineering. But I'm going to begin, and I'm going to spend most of my time talking about Dermograft and Transact, but I'm going to begin by just putting this in context in the development of tissue-engineered skin substitutes. So in the early days, in the 1950s and 1980s, sort of prehistory of this subject, all the tissue culture was pretty much all two-dimensional in monolayer culture. But during that time, there were developed methods for culturing fibroblasts and keratinocytes, which laid the groundwork for the subsequent development of the subject. So, three-dimensional cultures in this context go back to Eugene Bell in 1979, who put fibroblasts in a collagen gel and grew them in 3 dimensions. Now, there are several things that he noticed about these. First of all, he observed gel contraction. This may well be familiar to you. When fibroblasts are cast in a collagen gel, the fibroblasts contract the collagen gel by about 95%. At the same time, we now know that there's a great downregulation of genes. The cells downregulate collagen genes and proliferation. I'll show you that in a moment. And the cells become, the cultures become mechanical stress sensitive. So if we look at cell proliferation in monolayer, now this is looking at the rate of the mean growth rate in a monolayer culture, and this was done in a way using PKH26, where you could distinguish between different populations of cells. In monolayer culture, you find that all the cells are growing at quite a high rate. In a collagen gel, the majority of the cells are barely proliferating at all, and a minority of them are proliferating at a small rate. So in a contracting gel, you get a downregulation of growth, proliferation, and this is relieved somewhat if you prevent the cell, if you constrain the gel so that it can't contract. If you prevent contraction, then a larger proportion of the cells will proliferate, although a substantial number remain just not proliferating. So this is a system which has been exploited extensively, although it's not the system I'm going to discuss. But subsequent to that, Prunières, Bell seeded keratinocytes onto these gels, and they were able to make a stratified epithelium. In the early 1980s, Prunières realized that if you raised these cultures to the air-liquid interface, you could get much better differentiation of the keratinocytes, and you could get very skin-like morphology. You get very good skin-like morphology, and you could get cornification. And this looked very much like a skin substitute in vitro. Now, it's worth bearing in mind that, in fact, these keratinocytes are not differentiating in the way that they do normally in the skin. They are showing a wound-healing phenotype, where they display a lot of keratin 6 and keratin 16, which are not seen in normal skin at all. Now this kind of system has been used extensively to make test kits. Matec, L'Oreal, SkinEthic, and Genex are just 4 examples of this. There have been a lot of them, both academic and companies. Genex is actually interesting in that rather than making up a collagen gel and putting keratinocytes on it, they mix up the keratinocytes and the fibroblasts all as one big mess, and leave them to sort it out. And the cells do, in fact, sort themselves out so that you get a dermis and an epidermis. As these systems have been developed, particularly in the 90s, there have been a whole series of combinations of keratinocytes and fibroblasts, usually using fibroblasts and gels. Some of these have used things like vaginal epithelial cells. Some of them have been just grown a short time, or the keratinocytes have just been grown a short time so that you get more or less a monolayer, which is rather like a corneal epithelium, and it's been used for the kind of testing that is otherwise done with the Dray's test on the eyes of rabbits. But also cells like melanocytes and Langerhans cells have been added to these cultures. So these cultures have got really quite sophisticated. And they're used for testing things like corrosivity of detergents and some of these household products, some of the things that are, everything that's put in, all the cleaning fluids and everything else has to be tested, that it's not going to be corrosive to the skin, and that's performed by measuring NPT reductase activity before and after treatment, and you can look at the remaining viability of the cells. And also for testing sunscreens using UV light, which is another toxic agent. It's been used for looking for irritancy of cosmetics and things of that kind by the production of IL-1 or PGE2 after treatment. For looking at permeability. The permeability of these structures is about 10 times that of normal skin, so they're not actually very good. This probably relates to the poor differentiation of these products, but it has been used for permeability, and it's also been used for disease models, things like xeroderma pigmentosa. Now, this system is the basis of the product produced by organogenesis. This was graft skin in the early days, Aprigraph, which is a therapeutic product now, and they're used both for testing and therapeutically. But none of these systems show normal keratinocyte differentiation as you get in normal skin. And the closest to that I've seen actually is very recent, was a talk by Norbert Fusenek just a few months ago, in fact. It's the first time I've seen anything that even approaches normal skin in one of these culture systems. On the other side, there's some very interesting work from Adirans who've taken bulb fibroblasts with keratinocytes, injected them into mice or into these collagen gel systems, and they have observed the complete synthesis, complete elaboration of the hair follicle, complete with sebaceous gland and erector pili muscle. I mean, the whole thing seems to come together. If you're interested, that's a paper by Sten in the May Journal of Investigative Dermatology. However, what I want to discuss, what I'm going to spend most of my time discussing, is another system which is based on a really different principle. This was Gail Norton and Brian Norton in 1988, who developed a system which is now very familiar to you, of growing cells on scaffolds. In this case, the cells are human neonatal fibroblasts, and they grow in 3 dimensions on these scaffolds, lay down a lot of extracellular matrix, and wind up producing a tissue that looks, producing a tissue. And from this process, a number of products have been produced. Early on, ATS, Advanced Tissue Sciences, was making a test system rather like the ones that I've discussed earlier, based on this kind of dermal equivalent, dermal substitute, and it was used for testing corrosivity. It uses an ion scaffold, and in this particular case, keratinocytes were included. The 2 products I'm going to talk about are Dermograft and Transite. But these systems produce huge amounts of collagen. We produce collagen, human collagen, by the kilogram. And this has been used for cosmetic surgery, for injecting wrinkles and things of that kind. And we also, the conditioned medium was sold as a cosmetic, is sold as a cosmetic for skin treatment. And the point about these particular things is that to make a company go, you really try to exploit every possible aspect of what your material might do. So this is Dermograft. It's used for treating chronic wounds. It uses a degradable scaffold, which I'll discuss a little further, and it is cryopreserved at the end. And in tomorrow's talk, I'll say more about cryopreservation and its importance for this kind of product. Now, Dermograft is a therapeutic product, and so it's tightly regulated, in our case by the FDA, but also by other regulatory authorities. And part of producing a product of this kind is a very carefully defined, very carefully documented design control system, design system, to produce a design master file which goes to the FDA, device master file. So that begins with a product concept. And this was the product concept for Dermograft. So Dermograft was originally thought of as a dermal replacement. And in fact, it was originally developed for treatment of burns. Now that in fact didn't work out particularly well, so it was later seen as a cytokine factory. It was always going to be allogeneic. It was to be a viable product with viable cells in it. It needed a long shelf life, as Bob mentioned yesterday and on Monday, that there would be no sterile fill, because it was very difficult to imagine at that time. It's still difficult to imagine how you do a sterile fill with a live product. It is possible, but it's difficult. And then there's various things which are concerned with making the product usable by people by physicians, by people who use it. So this is the history of the product. This is how it came about. It was first a dream in somebody's eye in 1991. The development of cryostorage started very early. In 1994, there was a system developed, a scalable system suitable for mass production of this material using a hard bioreactor. That process didn't work. And in 1994, we returned to a system, not quite this system, but another bag system for clinical trials. And this is the system that we've actually been using ever since. And I'll talk about that tomorrow. In 1994, there was a pivotal clinical trial. There was, halfway through this, it turned out to be necessary to reevaluate the data and the specifications for Dermograft. That meant an additional clinical trial. We submitted a PMA. The FDA didn't like it, so there was another clinical trial in 1998, and finally the thing was launched in 2002. So there are various things that come out of this. One is that a lot of this process was being considered very early in the development. People got off the mark very quickly. There were false starts, like the 60-unit system, where people tried to do something. The only thing that remains of the 60-unit system are some files on my bookshelf at La Jolla. And the other thing is the false starts that you can get into with clinical trials. You really they need to be very clean. So looking at this process, the process uses human neonatal dermal fibroblasts. It is extensively tested, and then this essentially is an outline of the process. So the cells are expanded to passage 3 and cryopreserved at that point. That's treated as a master cell bank, and that in fact we have used only one master cell bank for 15 years, which is stored at passage 3. About 2 or 3 times a year, vials are taken from this and they're expanded from passage 3 to passage 5 and cryopreserved again. And this is known as the manufacturer's working cell bank. And then 2 or 4 times a week, vials from this bank are expanded again from passage 5 to passage 8 and seeded to the 3-dimensional systems. So this is how the system looks. Starting from the manufacturer's working cell bank, it takes about 5 weeks to perform the expansion, Seeding takes a day, 16 to 17 days for the growth process in three-dimensional systems, and then there's QC testing and release, which takes another 5 weeks. So the whole process is about 12 to 13 weeks. We'll come back to the significance of that, actually, in the talk tomorrow. The scaffold for dermograft is a PGA scaffold, PLGA scaffold, familiar to you, it's micro. It has interstitial diamond-shaped spaces in it, of this about 250 to 350 microns. And it is irradiated and it hydrolyzes actually quite rapidly, and I'll come back to that. By contrast, Transite, which I'll discuss a little later, uses a nylon scaffold which is not degradable, and it uses a celeste backing. So the PLGA scaffold, the strength of the scaffold decays quite rapidly. This is the harvest of the three-dimensional system, time of harvest of the three-dimensional systems, and really you can't measure the strength of this thing beyond about 3 or 4 days after harvest. And the material actually breaks down. Bulk of the material disappears about a week or so later. But the point here is that the tensile strength of this material is decaying as we make it, and there's very little left by the time we implant it. And this is how the three-dimensional growth process looks. Shortly after seeding, you can see the cells on the scaffold. They grow for a couple of days or so. By 8 days, and I'm not sure whether people can see this, you do see these interstices filling in. So we're getting three-dimensional growth at that point. And this is what the material looks like, and I hope this is visible. You can see the remains of the scaffold at the side here, and a great deal of extracellular matrix, which has been laid down by the cells. You can see the nuclei of them remaining within the structure. And we had a good deal of discussion as to what this material is, whether it's fetal or whether it's, well, it is a provisional matrix, there's no question about that. But the nature of the provisional matrix, and I think probably the best way to think about it is as a foreign body reaction occurring in vitro. So this is very similar to the material of a foreign body capsule. And when implanted into diabetic foot ulcers, this material aids the healing. This is a control. This is looking at this time to 100% healing. And we've got about a 20% improvement over the control at this stage. And that actually, the last implant was here. And this continues, this benefit continues after the last implant. Now, when it comes to marketing this material, selling it, the company has to be able to sell its product. A great deal of effort has been put into looking at the mechanism of action of this material. And from my point of view, this has been a large part of the development of this material. So we've always considered the mode of action of this material as a combination of functions of the material, and the emphasis has sort of developed as time has gone on. So at various times we've emphasized some of these more than others, but we consider the overall mode of action to be a combination of all these things. So the first point is that the cells in dermograft, many of them, about 50 to 80% of them, are viable, and they do in fact colonize the wound bed, and they stay in the wound bed. And they stay there for at least 6 months. And I'll discuss that tomorrow. They not only colonize the wound bed, but they also may recruit stem cells. They produce SDF1 alpha, stromal-derived factor 1 alpha, and this is macrophage chemotactic protein. Both of these are capable of recruiting stem cells, so you may get additional benefit from recruiting the patient's own cells. They produce some growth factors, and we've emphasized the androgenic factors, and I'll have to say a little more about that. But these are live cells, so they're also responsive to the conditions in the wound, in the chronic wound. Dermograft is also a good substrate for reepithelialization, and it modifies the inflammatory process. This is something we didn't emphasize early on because we didn't understand it. As time's gone by, we've come to understand that better, and I'll say a little more about it. So this is looking at the growth factors that we detect on a chip, on an Affymetrix chip, and actually an Illumina chip in this case. This is some sort of idea of what this tissue is making and what kinds of things it's interested in. I put out 4 groups here. Some factors involved with proliferation here. A set of miscellaneous factors here. There's a quorum sensor, a paraffin factor, and various factors involved with the synthesis of extracellular matrix. But the 2 biggest groups are angiogenic factors and these modifiers of the innate immune system, these inflammatory cytokines. So we start with angiogenesis. We've looked at angiogenesis right through from bench to bedside, as it were. So looking at the production of VEGF, this is, we're getting about something of the order of a nanogram of VEGF produced by dermograft per million cells per day. It's on the order of a nanogram per million cells per day. And that's a number to bear in mind, because I'll contrast that with some of the other things we found. We also have bound growth factors in here. I'm just concentrating on VEGF and FGF2. FGF2 we couldn't find, and we couldn't find it in the medium in early experiments. And the reason we couldn't find it was that it was all bound to the extracellular matrix, and we could only see it when we did a salt extraction. But actually there's quite a lot of FGF2 present in this material. We've looked at ex vivo. This is a standard angiogenesis assay using the chitotoic membrane. This is a control, and this is with dermograft, and we're looking for the production of these blood vessels here. And you can quantitate this in various ways. We've quantitated it in this case by counting counting branches, and you can see a stimulation of branch point production with the 3D cultures. This is dermograft, and we were able to block that largely with anti-VEGF, to some extent with anti-HGF, but a great deal of the angiogenic activity in this particular assay is due to VEGF. We've also taken this into the clinic. This is laser Doppler, looking at laser Doppler changes in blood flow in a diabetic foot ulcer, so this is the ulcer. And we're interested in the blood flow in this region. And as you can see, after 2 weeks it's increased, and after 5 weeks it's increased more. So we are getting an angiogenic effect right through from seeing angiogenic factors to seeing actual angiogenesis. And this is just to illustrate the fact that we've got a good substrate for keratinocyte migration. This is a piece of skin on a piece of transite in this particular case. And these keratinocytes have epithelialized the piece of skin. And when they've reached this point, they've elected to move out on the transite rather than continue epithelializing the piece of skin. So they prefer transite as a migration substrate to a piece of skin. And in fact, this substrate, this extracellular matrix, is a complex mixture of various proteins. If you contrast this for a moment with the collagen gel assay, in a collagen gel, you put the fibroblasts into collagen gel, and it's like shouting collagen at them. And they respond to collagen. This material is much more complex. So it contains some collagen. In fact, it contains several collagens, 5, 6, and 3. But it contains also fibronectin, tenacin, which will modify the fibronectin, various small proteoglycans, deca and lumican by glycan, and it's a much more complex message to the cells. It's more like the cells getting a page of text to read, rather than just being shouted at. And they respond correspondingly. Okay, so looking at how at a chronic wound, it's nice to have some idea of what you're trying to do when you're treating a chronic wound. And for that, there are a number of hypotheses that have been put up based on information protease and so on, keratinocyte defect. The particular one I'm going to discuss this morning is concerned with fibroblast senescence, because the fibroblasts that are isolated from chronic wounds do show senescence-like phenotype and consequent bacterial colonization of the wound bed. So this is the hypothesis. Fibroblasts show senescence-like phenotype. They're becoming unresponsive to stimuli, and in particular they're unresponsive to injury or unresponsive to bacterial colonization, and as a result, since they don't respond appropriately, colonization occurs, and that prevents closure of the chronic wound. Now, the fibroblasts are senescent, and there are a whole lot of reasons that they might become senescent. ROS is the one I'm emphasizing here, and you get ROS from a variety of sources: ischemia, reperfusion injury, iron from extravasation of red cells in the case of venous stasis ulcers, high mitochondrial potential in diabetic ulcers. And the ROS will damage proteins so that they don't break down properly, will damage lipids which will interfere with signal transduction, and attack DNA, which will cause cell cycle arrest. When we look at what we've done here is to look at what happens to the inflammatory phenotype of fibroblasts as cells approach senescence. And we've used replicative senescence in this particular case as a surrogate for doing other things. And as you can see, the cells become more, this is looking at IL-8 production, just again as one particular cytokine. The cells become more inflammatory as time goes by, but they also become much more variable. And this looks like a dysregulation of the response of the cells. In fact, if we, as the cells age, as the cells get out here towards becoming senescent out here, their growth rate is declining. And if we plot IL-8 or other cytokine production against growth rates, then we can find some correlations, but you're getting an awful lot of, it's reasonably consistent down here while the cells are growing rapidly, but when the cells start growing slowly, you get a lot of inconsistency, a lot of variability, and eventually there's actually a drop in the production of IL-8, and we see this with IL-6 also. And we get a lot of cultures which just produce no IL-8 at all at this stage. And the argument here is that IL-8 is concerned with bringing in bactericidal activity. So out here we're getting patches, we're getting cultures here, but we can infer we would get patches in an ulcer where you would get no bactericidal activity and bacterial colonization could occur. So this is looking at some of these cytokines. These are the ones that are highly produced by fibroblasts in three-dimensional culture. I've just been talking about IL-8, but these are all very similar. They're secreted by a whole variety of cells, and they are chemoattractants and activators for neutrophils. They have two receptors, a high affinity receptor, which is concerned with bringing neutrophils in, and a second receptor, CXCR1, which is the low affinity receptor, which is concerned with activating the neutrophils when they arrive. So these cytokines are concerned with bringing in neutrophils and activating them to a bactericidal activity, and that is something that seems to be dysregulated in the fibroblasts that you see in chronic wounds. So this is the hypothesis. The fibroblasts with this kind of senescence, it's called stress-induced premature senescence, the way I've described it this stage, fail to produce IL-8 and the other cytokines, so the neutrophils are not brought in, and bacterial colonization occurs. And the argument here is that keratinocyte migration is delayed by bacterial products. It seems sensible. You don't really want to close a wound over live bacteria. And in fact, we know of mechanisms for stopping keratinocyte migration. Neutrophils produce an elastase, which breaks down fibronectin and causes discourages migration of the keratinocyte. But we were wondering whether there was any direct effect where the keratinocytes themselves could detect bacteria. So what we did was a scratch assay in which you take a lawn of keratinocytes, make a scratch, and the keratinocytes will migrate to fill in the wound. So this is what you see. So you've got a scratch, and the keratinocytes migrate in to fill the wound. If we add lipopolysaccharide, which is a bacterial product, and strictly this is the bacterial product, you can, if you're close enough, you can see that these cells are actually doing things. They're putting out pseudopods and jumping around and one thing and another, but they are not moving. So the bacterial products will just stop keratinocyte migration dead in its tracks. And we've been able to show there's a dose response to this, and that the keratinocytes are detecting the bacterial products through toll-like receptors TLR2 and TLR4. So this is how it works. This seems to work. So what does Dermagraft do in all this? Well, one of the things that's very striking in a three-dimensional culture of fibroblasts, you compare three-dimensional fibroblasts with two-dimensional fibroblasts, you see a dramatic upregulation of these chemokines, NS78, GCP2, IL8, and GRO-alpha. These are the ones that we've been discussing. So there's a dramatic upregulation, and there are a series of other things that happen. And so the 3D cultures that we're using here and implanting induce these agents. And in fact, they produce large quantities of them. This is just looking again at IL8. And we're looking here at 30 to 50 nanograms of IL8 made per day per million cells. Remember, VEGF is about a nanogram per mil per day. This is between 1 and 2 orders of magnitude higher than the amount of VEGF. There's a great deal of this chemokine produced, which will bring in the neutrophils, activate the neutrophils, and provide an antibacterial activity, which will clear residual bacteria. So the thought here is it's probably a high efficiency rather than a high capacity system. So you need to debride to get rid of bacteria and use antibacterial treatments to get it down as low as you can. And then this system is good for cleaning out the last few bacteria and allowing keratinocyte migration. What I emphasized at the beginning is that this is the activity of Dermagraft is a combination of all these things. So that this yellow path through here is what happens in acute wound healing, which has been arrested in an ulcer. And Dermagraft provides cells which will provide a substrate for migration. It will provide various growth factors, some angiogenic growth factors, and also growth factors, cytokines, growth factors and cytokines, which will influence the immune system, as I've just been discussing. So just to say a few words about Transite. Transite was designed for treating burns, for severe burns. So this was the original product concept. It was a transitional covering for burns to replace cadaveric skin. When a burn is brought in, the physicians will know you need to remove the eschar as quickly as possible, and you have to cover the patient with something. The standard has been cadaveric skin, and it's probably going to be again, but cadaveric skin is rejected within about 14 days. So you have to get the patient stable and everything else within 14 days. Transite, we've had it on the patient for 120 days without any trouble. It's an allogeneic product. It is non-viable because we found that viability didn't help. There's a long shelf life because you never know when you're going to need these things, and then there's a series of things concerned with being convenient to the physician. So this was a concept in 1993. We tried the 60-unit system. We had the same sort of experience as we did with Dermagraft. It didn't work. I mean, we tried it and it didn't work. There were a series of preclinical studies in 1994, and this was where we found that the non-viable product was better than the viable product. It was, we were not getting good results with a viable product. So we approached the FDA in 1994, did a small clinical trial, and we had the thing on the market by 1997, which is extraordinarily fast for any of these things. And in 1998, the use was extended to second-degree burns. Now, yes, we're not going to be able to see this. This looks very much like what you see dermographs, that you can't see it. I'll concentrate on that. The production process for transite is very similar to the production process for dermograft. So this is a matter of capitalizing on what you're already doing. So the expansion process is the same process for both dermograft and transite, and then we use a somewhat different bioreactor for the second stage. So there are minimal differences between these two processes. Okay, so how does transite work? Well, this is what we said about dermograft. And some of these functions depend on live cells, so none of those are going to apply. So you're left with just a few of these things. Growth factors, reepithelialization, and the modification of the inflammatory process, some parts of that. But in fact, there are other factors that are important for transite. As I mentioned, the extracellular matrix that's laid down is as much like a foreign body capsule as anything else. So we're making a foreign body capsule in vitro so that the patient doesn't need to. Now, if you just put a biomaterial onto a burn, the patient is going to make a foreign body capsule to that material, and that foreign body capsule is going to be part of the patient. And if, the material will grow into the patient. The other thing about this is that at the time of harvest, this production of a capsule has been going on for two weeks or a little more, right, a little over 2 weeks. So it's partially completed, it's no longer getting started, and it's therefore much less inflammatory. It's not getting the process going. So far as the properties are concerned, it doesn't contain angiogenic growth factors itself, but for reasons we haven't sorted out, it seems to be capable of stimulating production of, in this case, VEGF, by fibroblasts that are co-cultured with it. Even though it's non-viable, it can still cause the production of quite a substantial amount of VEGF from fibroblasts. It's used for second-degree burns. It reduces dehydration, it reduces pain. In a second-degree burn, there are plenty of keratinocytes, so the emphasis here is on keratinocyte migration. And it also allows lymphocytes to come in that prevents, leukocytes come in that prevents or reduces infection. I mean, it controls infection. In the case of third-degree burns, the major features of it are that it's non-immunogenic, and it also produces a cleavage plane. As I mentioned, it's a capsule that has already been made, and when you lay it onto a patient, the patient doesn't make a capsule themselves. So that, I hope you can see this. This is a piece of transite on a mouse. You can maybe be able to see the nylon scaffold in some of these places. This is mouse here, and the transite is this material here, and going through here is a cleavage plane. If we left out the preformed capsule, you would see the mouse, material forming a capsule which would be part of the mouse. And as a result, this material doesn't bind so tightly to the wound, and it can be removed just by pulling it. Now this was an experiment comparing the scaffold that we use for transite with transite itself. We put pieces of transite onto a nude mouse, left it for 2 weeks, and then tried to remove it. And you can see that the force required to remove the remove the scaffold alone is somewhat greater than, in fact significantly greater than it is for transite. So it appears that this pre-formation of a capsule does have a real effect. So, just looking at what we picked up from this, The production of these products starts with a design concept, which sets out what you plan this material to be, and from that feeds a whole series of design reviews and design stages, which right the way down through to the finished product. Various little bits become their own design project. There's an early evaluation of requirements, and that's important, and the important point here is that it's early, and I'll discuss that further tomorrow. An early exploration of production methods, like the 60-unit system, which didn't work, but it was explored early on. It could be discarded, and it could be discarded without having a major effect on the final product. If you discover later in the process that your production process just doesn't work, it's sort of difficult to deal with. The clinical trials and the re-evaluation that in this case had to occur. And the importance there is, importance in that case of doing the clinical trials properly, so that they're acceptable, so there's no questions from the FDA about this, because the FDA can be very difficult, or these regulatory authorities in general can be very, let's say, finicky about exactly how clinical trials are done. I haven't said a great deal about the launch and distribution, but I'll say a little more about that tomorrow. But the support for sales, a great deal of the effort that's gone into this material recently has been concerned with the support for sales and the mechanism of action. And then finally, the question of reimbursement and clinical tracking, which is something that is just... Reimbursement is a major question, certainly in the United States, with regard to the use of these things. If you can't get reimbursement, it's very difficult to sell these materials, and the clinical tracking is something that provides much better information on how the material is actually doing than you get from a somewhat limited clinical trial. Okay, that's the story.
So let's take some questions. Let me ask one, Jonathan. Dermagraft.
Mm-hmm.
Degradable scaffold.
Degradable scaffold.
Cryopreserved. Mm-hmm. Is the scaffold still to some extent present at the time it is cryopreserved?
Yes.
And if so, what happens to the degradation rate while it's cryopreserved?
We haven't worried about the degradation rate of the scaffold while it's being cryopreserved. The argument being that the degradation rate of the cells is substantially higher, and we actually determine shelf life on degradation of the cells, the slow degradation of the cells at -70. Degradation of the scaffold is likely to be much less than that, so that it's not an issue. The scaffold, in fact, only lasts for a few days after it's put into the patient. You can't find it. You cannot find dermagraft a week after it's been put into the patient. It's been absorbed into the patient.
Yes, back there.
I want to ask about future hopes about tissue engineering the cornea. Instead of taking corneas from the cadavers or from tissue banks, is there any future hope to tissue engineering the cornea?
The cornea. We made a corneal product for testing at one time. Now there are other groups that are using sheet techniques for producing corneal epithelium, and they're actually being very successful. What they do is they grow keratinocytes on a NIPAM surface with a mask it's a lifting device essentially, and then they cool the NIPAM and they can remove the sheet, and that has been used quite successfully as a grafting system. At this stage, it is still a laboratory, hospital kind of operation. I don't think anybody is doing it commercially, but it's certainly the technique has been worked out in principle to enable that to be done.
Until we are waiting for the commercial products from the Korean, why don't we use the xenografts for the corneal grafts instead of allografts?
Why don't we use
The xenografts from other species, as to the best of my knowledge, that cornea does not need any tissue matching.
Well, there are a couple of reasons for not using xenografts. One is that for a regulated product, the testing becomes more stringent, and you've always got, at least if the regulatory authorities have at the back of their mind, that there may be problems with zoonotic infections. And we haven't needed to. This product is only fibroblasts, and we have used a single donor. And those fibroblasts, the master cell banks, have lasted 15 years at this point. So we haven't needed to use animal tissue.
In using animal cells, there's a great concern, at least in the U.S., about the transfer of animal viruses into the human being. So,
Thank you very much.
Right.
did you have a question? Let's pass the microphone forward.
My name is Dr. Mustafa, Professor of Pathology in the National Cancer Institute, Cairo University. I really have two questions. The first one about the application of this sort of technique of alternative to skin grafting. You know, you are using it mainly in burns, and the underlying tissues are not very healthy in case of burns. But with tumors, after you remove the tumor, we usually use classic skin grafting systems. But sometimes we have deficiency in certain areas, so I was wondering whether this grafting system can work or not. The other question about the availability of these things in the market, especially in the Egyptian market. Thank you.
The availability
of this grafting.
Availability is a problem because Smith and Nephew has decided that it is not willing to do this anymore. So we're not able to produce it. Whether that will change in the future, I don't know. It'd be great if it could. We certainly were selling to Saudi Arabia and Thailand. I'm not sure into the Middle East, other than Saudi Arabia. But certainly, that is a question of availability. And we would certainly, we would have liked to have done it. Now the other question was
about the application to cancer.
Okay.
After treatment.
Right. Yes. There's been various thoughts about filling spaces. Now I think the problem with that is you need to get angiogenesis into it fairly quickly, or it's not going to survive. It will be possible to use a non-living material, non-viable like transite, but as a sort of bulk material. That would be fairly expensive to produce, but it could be done. Dermograft has been used for Mohs surgery in elderly people where the wound is very difficult to close. And it has been used successfully. It works quite nicely. My experience with Mohs surgery is that the feeling is that the patient just gets a facelift, and they don't really need anything to fill in the hole. But I think in some circumstances it could be very good. In that particular case, you would be getting migration of keratinocytes from the edge, because you would have taken out all the hair follicles and so on. And that takes a little while, but it would be possible to put in a dermograft, a piece of dead dermograft, should we say. And that is certainly a superior substrate for keratinocyte migration to the wound bed itself.
Okay, we're going to take one more question, and then we're going to move on. Okay, Steve Allen.
Jonathan, I think you said the shelf life was 6 months.
Yes.
For dermograft. That's 6 months from the end of that 13-week period.
No, in fact, it's 6 months from the time of cryopreservation, which is prior to QC testing, which takes 5 weeks. So we take, and I'll discuss this tomorrow, but we take 5 weeks out of that.
So you're really down to 5 months.
We're down to 5 months by the time we've got the stuff cleared.
And what was the what changes after 6 months that makes it no longer as good as it was at 6 months?
Okay, we do this on the basis of MTT, MTT reductase activity, which is a measure of metabolic activity, and it's inferred as a measure of viability,
So
whatever that is.
so
But that slowly declines at a rate of about 0.02 MTT units per month.
Even cryopreserved?
Yeah, -70, at -70. If you took it down below glass transition temperature, which is what, -120 or more, then you can stop that. But that has other practical difficulties.
Okay, I think we need to move on. Thank you, Jonathan. Next is Steve Badelight, continuing the discussion and talking a little bit about commercialization of SIS. Steve, introduction at this point. Do we have a microphone?
He took it with him. Should have left it there to see what it really was about.
Donald Smith and Nephew people are always trying to shut off other people talking.
Okay, is this volume okay? Thank you. So, Bob asked me to talk a little bit about the commercialization of this porcine-derived extracellular matrix that we call SIS, or small intestinal submucosa. And it's sort of an interesting story, at least it's interesting now. When you're going through it, it's a little bit different. So, this is actually the way this happened, as opposed to the way one would predict it. You know, normally when you're thinking about a concept or a tissue engineered product, you know, you get an idea and then you go into the lab and you test it and say, does this work, that work, whatever, and eventually you figure out the things that make something work and you take it to a clinical application. Then you go back and repeat things. Well, in the case of SIS, what we actually did was found that it worked for a particular application, which happened to be as a blood vessel graft, an aortic graft. We were in an entirely different experiment. We were using the entire intestine as a graft and then eventually figured out the layers that were making it work. And then we went from there to intellectual property protection and trying to figure out what's going on. And then we eventually said, you know, this must be because it's extracellular matrix that has all of these things. So it was sort of just the opposite. And then we went back and did some more R&D and more IP and then more applications, and the thing goes back and forth. So I'm not sure what's the right way to come up with something that works. But if you were making this chart from scratch, you probably would come up with concept first, and I said it'd be just the reverse. But then eventually you go back and forth because you learn as you go about the things that are working. So one of the things that is important in the development of a product, because what Dr. Mansbridge was just telling us about was from an industry perspective. Here's how you develop a product. Here's the issues, here's the problems, here's the way you commercialize. The problems you run into and the successes. I come from a university perspective, so the discovery that occurs in my laboratory, if it's ever going to see a patient, somebody's going to have to make money off of it. So we're going to have to partner with industry. And the sooner we recognize that, the better. And which means that the way we do research in the laboratory probably ought to be thinking about this very early, which means we ought to keep our laboratory notebooks, the way in which they'll withstand the scrutiny of a challenge later on the intellectual property. And we've got to get good intellectual property protection early, meaning patenting, disclosing potential inventions. But more importantly, then getting industry involved, because there's just simply no way that we're going to be able to do the type of development work that needs to be done to get an idea to a product stage on federal monies or grant monies. We need the type of monies that industry can provide and partnering with them. So this we learned relatively early. So the critical issues in partnering with industry, in my opinion, are, number one, there has to be an absolutely very strong commitment by whoever you need a champion within the company. And when we were working with SIS, we had several industry partners, but the first partner
that really made it go was a DePuy, which is now a Johnson & Johnson company. There was an individual in that company named Dick Tarr, who was the Vice President for Research and Development. He took ownership of this particular idea, and to show you how committed they were and all the things that could have gone wrong, in 1992, they licensed the intellectual property from our university, and he told the company it would be at least a 7-year product development scheme for what he had in mind. And in the meantime, there were 3 changes of ownership of a company that had been private for 100 years. And now the things that can go wrong when 3 changes in ownership occur are just tremendous. I mean, they could drop certain initiatives, raise it, but he stuck with it. And by the time the 3rd owner, which was J&J, came along, there was $16 million invested in the project. Everything was on track, but here's a new industry owner coming in and saying, wait a minute, you got here $16 million in the red, you know, where's the product? And he said, well, I've never said we'd have a product at this point, but if you hang in there, and he talked them into it, and eventually, in another 2 years, they ended up treating it, and they're well into the black now, but without that strong commitment by an individual in the company, it never would have happened. The company is also the ones that helps develop a regulatory strategy, because even though we think we're learning how to do this in universities, really it's industry who understands how to work with the regulatory agencies, and they also got the manpower to make that happen. The issue that was addressed by Jonathan too on reimbursement for tissue engineered medical products is something we can't manage from within a university. That has got to happen from an industry perspective. The lobbying groups and the partnering between industries that goes forward to the agencies, in all the different countries. You know, it's bad enough in one country, the United States, but then you have to go to each country and figure out what their reimbursement issues are. So there's no way that that's going to happen either without a strong industry partner, because they're the experts at this. And then where the partnership really becomes important is in education, because the concepts of whatever product it is. In our case, it was an acellular scaffold taken from a pig. And the real understanding of it resided still within our laboratory. And this happened to be an orthopedic company that was this first licensee that was used to working with metals. Orthopedic companies in the early 90s were almost exclusively hardware companies. And so now you're taking a sales force and an R&D force within a company that's used to thinking from a very engineering perspective on how to make devices and how to sterilize them, and educating them about biology. And so we met with them, our entire research team met with their entire research team every 6 weeks to start with, and then eventually it stretched out to every 12 weeks, but that occurred over a period of about 5 years. We would take our whole team up to their place, they would take their whole team down to our place, and we truly worked back and forth. What I'm describing to you is an unusual situation in the way universities work with industry. And maybe we were just lucky in the way it worked, or maybe it just happened to be the people that were involved. But I think it is the reason that it worked, and I think it's also the lack of those things are the reason that they fail. This education of the sales force is really important too, because education of the physicians is just as important. If you recall 2 days ago, one of the graphs that I put on the screen here was the typical response to injury. You get injury, you get hemorrhage, you get inflammatory cells, and all the things that happen ending up with scar tissue formation. And so surgeons, who are the main users of tissue engineered products, are very well trained. They spend 12 to 15 years becoming trained and understanding why certain things happen and how long it takes to happen. If we stick with just one particular example, orthopedic surgeons, for example, say, okay, if you break your arm, I put a cast on there. The cast is supposed to come off after 4 weeks, and we do a rehabilitation protocol that has been time tested for 15, 20, 50 years. And why should I change? That's simply the way things happen. Or if you have, let's take a soft tissue example of an Achilles tendon rupture. If you put a gap in an Achilles tendon, the conventional thinking is that this is going to be an extraordinarily difficult fix, that it may never get fixed. At best, you're hoping that the patient is, a year from now, going to be able to get back and be walking reasonably normally. So you gently bring these ends back together, you protect them with a brace, you gradually you leave them a fixation on for 4 months, and you gradually load it, and eventually you hope that this thing doesn't break. And so we come along with a tissue engineered product, and we say, stick this in here, totally change the way you do surgery and rehabilitation, trust me, it'll work. Well, they don't, because they've been taught for 12 or 15 years that this simply doesn't happen. So perhaps education of the users, the eventual users, is even more difficult than education of the sales force. So these, I think, are critical issues in a university working with industry to take a tissue engineered product out of a laboratory to a patient. So let me mention just a few commercialization issues as well. I think the critical issues here involve the type of license agreements that occur between academia and industry. And I don't know what it's like in Egypt, but in the United States there's significant room for improvement. And there are lots of reasons for this. But the license agreements that occur and the terms that go into that, basically they set the stage for the partnership. If you've got one group of people trying to do a license agreement, but it's another group of people who have to make the partnership work, and those two groups of people don't talk to each other very closely, there's significant potential for problems, and those happen quite frequently, at least in the States. The other things that have to happen is, in my opinion, like I said, the expertise for the particular invention, and all of you have got different inventions, resides in your laboratory. So when the license agreement happens and you're going to take this idea and try to commercialize it, it's very important that this relationship between the inventors in the laboratory and the industry representatives is at least good enough so that there's a good exchange of ideas and cross-fertilization. It almost never happens that the product is commercialized the way the inventor originally wanted, but that's okay as long as there's communication that's going on back and forth, because if not, then there's going to be some real problems. And finally, one of the critical issues, in my opinion, in commercialization is that you're in your laboratory working, and you have industry out there commercializing an idea that you had several years ago and developed in your laboratory. Well, you're not going to stop working. You're going to keep doing what you're doing in your laboratory, and you're going to be trying to make it better. So you're going to be trying to invent or discover a second or a third or a fourth generation product that beats the first generation product. Now, if there's not an agreement in the light, some understanding between the university and whoever you've licensed the first generation product to, now you've got real problems, because 3 years later, you've now invented something that maybe makes your first invention obsolete. Now think of what happens to the relationship between the university and the industry when they supposedly are going to be working together, and now you're coming along with an entirely new invention. And unless there's some way to handle that in the agreement between the university and the industry, again, there's real room for problems there. How do you allow your first partner to take advantage of what continues to happen in your laboratory, because that takes money? Or do you say, well, sorry, now we're going to go license to your competitor something that's, you paid a lot of money for 3 years ago. So I'm raising these issues that are very real and that perhaps you don't think about until you've been through them. But this is from the university perspective, which gives you a little bit of a different idea on it than what Jonathan told you. I already talked about the education aspect of this. Now let me talk a little bit about this particular invention and eventual product, small intestinal submucosa. And in my laboratory, the only time we use SIS anymore is as a control, because everything we've been doing in my laboratory for the past 10 to 12 years has involved this second and third generation work on and an understanding of why SIS was working. Basically, we're trying to understand the mechanisms. Similar to what Jonathan said at the end, understanding the mechanisms of how things work are very important to a sales force, because you can't simply give something to a physician and say, trust me, this works. They're going to say, show me it works. Show me why it works. I need to understand this before I'm going to pay X number of dollars for it. So here's the small intestine of a pig. Everything in red on the inside, here's the lumen or the middle of the intestine, it is the mucosa. This blue part's the submucosa, and out here's the muscle layer. So the muscle layer gets removed, the mucosa layer gets removed, and we're left with this material that is the middle part. This connective tissue is the extracellular matrix. And just to give you a little bit of a reminder of a histology lesson, there's a lot of layers to the intestine. If you consider this part up at the top, the lumen or the middle of the intestine, and down at the bottom would be outside of the wall of the intestine, or the serosa, you go from the epithelial cells through the basement membrane that they sit on, the lamina propria, and then the basilar portion of the lamina propria is compacted down, and it's been given the name stratum compactum. And then there's a muscularis mucosa, the submucosa, muscularis externa, and the serosa. SIS only consists of the submucosa and very small amounts of the muscularis mucosa and stratum compactum. It's a trilaminate structure. That's what the patent is based upon. And so that white material that you saw is these three layers out of the intestine. Now, this might seem like trivia to you, but it was absolutely essential to gaining the intellectual property protection for this particular product, because submucosa alone had been tried for a long time and had gotten entirely different results by people back as far as 1938. So again, here's a histologic section. Over here, the mucosa has been removed. This dense layer is the stratum compactum. You can't even see the muscularis mucosa, and then the submucosa here. So this area right in these three layers are SIS, and then the muscularis mucosa gets removed as well. So now, here's what we did not think about early, but going back later seemed to make a lot of sense. So what does this layer, this SIS layer in the yellow here, do in nature? Well, it supports the most rapidly dividing cell population in the body. So every 5 days, the entire intestinal epithelium turns over. So this must be a very good substrate for cell growth to support that sort of a cell turnover. In addition, it has to heal very rapidly. If we don't heal our intestines very rapidly, we die. You get injury to your intestines, it better seal up quickly, otherwise things are going to leak out or it's going to break open into a wound and we die. So perhaps one of the evolutionary phenomena that's happened is we've got a very efficient wound healing mechanism in this particular area of our body. We can afford to delay healing for a little bit in most musculotendinous tissues, but not in the intestine. So we've got a rapid wound healing, and in addition, it has to heal without scarring, because what happens if the intestine scars? You get stricture, and you might as well not heal, then food doesn't pass. So now you've got an area that supports rapid cell population, rapid wound healing, minimal scarring, and it also has got a function for somehow processing all of the different immune antigens, all the antigens that pass it. All the different things we eat have got to be processed, but by the time they end up in our system, they're no longer immunogenic. So that's really a black bag in there. And finally, it's got to have the mechanical strength to withstand all of the things that happen. It's got to push food along. It's going to have all different sorts of shapes and sizes and consistencies, and it's got to be able to withstand those. So this is sort of phylogenetically and physiologically what happens in there. So you think, God, that ought to make a very good substrate. That ought to make a very good scaffold for tissue engineering. Well, after the fact, all those things seem to be obvious, but we certainly did not think of those things early on. So then you start to ask, well, what's most important? Why does this stuff work like it works? I'm going to show you a couple of examples in a minute here. Is the composition of the material important? Or is the structure of the material important? Or is it some combination of them? And so we've been spending, I don't have the answer for you. We've been spending 15 years at least trying to understand that. I'm guessing that it's a combination of these things, but I don't know which is more important. I can tell you, though, that we spent a lot of time understanding the composition of SIS, just as ATS has spent a lot of time and money understanding the composition of dermagraft. What is dermagraft? I mean, dermagraft is extracellular matrix produced in vitro. SIS is extracellular matrix produced in vivo. So there's probably a lot more influences on SIS production because you've got all this other stuff going on around it and probably affecting cell phenotype and differentiation, and it's very controlled in the dermograph situation. But we know for sure that this list is going to look familiar to you, to dermograph. There's collagens 1, 3, 4, 5, and 6 in SIS. We know these for sure. There's at least 5 glycosaminoglycans, chondroitin sulfates A and B, hyaluronic acid, heparin, and heparin sulfate. And we quantify these things. We know how much of each of them there are. We know that there's glycoproteins like fibronectin and tactin and laminin in it. There's proteoglycans, decorin, biglycan. And the following growth factors is only a partial list. VEGF, TGF-beta, EGF, IGF, SDF1, there's an NGF-like factor, basic FGF, and the list goes on. And we quantified all these things. So even though we know all of these things and how much there is, if we take all of them and we throw them into a test tube and we try to recreate extracellular matrix, we never get as good a result as if we harvest it from nature. So I spent some time the other day trying to explain to you why processing and manufacturing is important, because we have been unable to recreate the type of a scaffold that basically Mother Nature's been millions of years of R&D at this, right? We've got a scaffold that has evolved over time to be very efficient for a particular use and a particular set of applications. So this is what it looks like. That's what it looks like under a scanning EM, and that's what it looks like if you make particles out of it and compact them together. And this is the collagen architecture of it with little proteoglycans, little globular proteins stuck to their surface. And of course, there's no holes like this in nature. This is an artifact of the preparation to get this picture. It's a salt gel, it's a solid, and so when cells go through it, they migrate, they break down the matrix ahead of them and then migrate through it. Now, this looks like a random orientation of collagen fibers, but I will bet my car that that's not true. I'll bet you there's a rationale for that, and that if you look at every different tissue or even different parts of the same tissue, the organization of those collagen fibers has a purpose. If we understood that, then we could probably get closer to recreating it in vitro than we can now. Because we can solubilize collagen and repolymerize it, probably, with electrospinning now, we can, make it into almost any shape. We could probably recreate this orientation if we understood what it was. It takes a lot more computing power than we have, that's for sure. So here's some more on structure. I've given you the composition and some evidence of what structure looks like. Remember yesterday, this is from the urinary bladder matrix. Here's the basement membrane up on the surface and the underlying tunica propria. This is SIS. Here's that stratum compactum layer up here, and down below it is the less organized submucosa. And this is the extracellular matrix from the liver, a completely different architecture altogether. That's because each of them has a specialized function. One supports liver cells, one supports the different layers of the intestine, and the other one supports the smooth muscle cells and has to keep urine in the lumen. So they've got entirely different functions and therefore entirely different structures. So you can get creative with this and make a lot of different sizes and shapes. And we looked at some of these the other. You can do this. You can make any of these sizes and shapes from any extracellular matrix. And the only reason we do this is for clinical applications. We make a gel form because we can inject it. Now, if you make a gel form, you've solubilized it, you've got all the composition there, but when it repolymerizes, you've totally lost the structure. Whereas everything else still has the native ultrastructure, it's just that these are very small particles, still the same ultrastructure. This is 10 layers, this is 4 layers, this is 2. They are still all SIS. And so when the body sees them and cells migrate and attach to them, they see the same surface architecture. So this is, I'm not going to go through this for urinary bladder, but other than this, it's got the same number of layers just about, and that UBM was just the basement membrane, this very thin layer here in the underlying tunica propria. We throw away everything else. But it looks very similar macroscopically. All right, so here's some of the things that we've done with at least 2 extracellular matrix scaffolds, SIS and the urinary bladder. It's been commercialized now for cardiovascular, musculoskeletal, gastrointestinal, lower urinary tract, neurologic, dermatologic. There's about 20 FDA allowances, 17 for SIS, 3 for urinary bladder. been about 350,000 human patients implanted, and there's more than 60 worldwide patents protecting this intellectual property. How can you get 60 patents on a piece of intestine? It's just amazing. I mean, you can one thing you learn is you can patent anything. If you've got enough money and you go and enough time, you can always get something out of it. And that becomes important to your industry partners, of course. So we've been very lucky, very fortunate with the commercialization of these extracellular matrix biologic scaffolds. Okay, so a couple of clinical applications here. And now think of this. This is a xenogenic, a porcine-derived extracellular matrix being put into some different species. And most of what I'm going to show you is human. And a lot of different applications, even though we're taking it from, and I just got done telling you that how specialized these ECMs are for each location, they still work well enough that we can take an intestinal submucosa and put it in places like an Achilles tendon. This is an Achilles, there's a huge gap in the Achilles tendon of this woman. This was done by Dr. McLeod in Atlanta, Georgia. There's a heel of a patient, foot goes down this way, leg comes up, 6 centimeter gap. Now if the surgeon pulls this together, the heel is going to be lifted off the ground, and there's so much tension on it, it probably wouldn't last. So this is about as close as he can get it. Closes it to about 3 centimeters, which is a very big gap. He takes a piece of this 10-layer SIS and tubularizes it, and fills the gap like that, and overlaps the adjacent normal tendon. Now the key was that this patient then was not put in a cast. Any surgeon would normally put this patient in a cast because they would know that it would break otherwise. So this patient instead was put in what's called a walking boot, which allows them to bear weight immediately. And every night we said, take that boot off and hand your leg over a table so there's no weight bearing, keep your range of motion going well. And then at 4 weeks, even the walking boot was taken off and the patient was allowed ambulation without external fixation, which is so different than the way that orthopedic surgeons would normally handle this, that you'd almost set yourself up for a lawsuit in the United States by doing that. So we've got about 185 patients in this series now, all of them treated identical, and not a single patient has failed yet. And at the end of the day, what happens is that the ECM scaffold turns into this. These fibroblasts have now secreted an appropriate type of load-bearing collagen that is aligned along the lines of stress. This is from distal up to proximal, and you can see that they all line up appropriately. And there's no SIS left. The SIS is gone after about 6 weeks, just like the dermograft. You say you can't see it after, what was it, a week, Jonathan? Macroscopically, you can't, probably there longer, but this scaffold disappears extremely rapidly. This is an interesting patient. This is from Long Island, New York. And this patient had 7 years failed treatment for a post-deep venous thrombosis problem. 10 surgical procedures, aplograft was used, vein grafts, skin grafts, and this picture was taken after the surgeons actually tried this orthopedic SIS device. The history is they didn't get a first picture, that from about here up to just below the knee, 3/4 of the circumference of the leg had bone exposed. And this patient's leg was so chronically infected that he was relegated to the corner of a house in the back room because it smelled so bad. And the quality of life for this patient was awful. And so he was willing to try anything. So we took these layers of SIS, laid it across the bone, and very rapidly filled it in. And so this is about 6 weeks after the initial treatment, and it sort of stopped here. And then we did another treatment, and it looked like this after 3 more weeks. And then in 6 weeks, it looked like this. So you can see even the size of the leg decreased, the swelling decreased, and it's got an epithelial covering. And even though this isn't anything like a normal leg or normal skin, this patient now's quality of life has changed. Remember the definition we talked about the other day? One of the things that I believe we should be thinking about in regenerative medicine is, if we think of it as changing the default mechanism of wound healing, so that instead of not healing or healing with scar tissue, we can heal with a more constructive type of remodeling. Even if we don't achieve total regeneration, we'll keep trying to do that. If we can change the default mechanism, then I think we can do some good. This is not a person, obviously. In fact, you might not even know what it is. This is the face of a horse. Here's an eye, huge hole in the face of this horse. And I got a call from a veterinarian at Colorado State University because this horse had been like this for 2 years, had stopped healing, and this goes straight into the nasal cavity. The interesting thing is you can see there's not really a lot of infection there. It's actually relatively healthy tissue. There's a huge hole in the base of this horse. So I went out, I happened to be in Colorado and stopped in and saw this, and I mean, the horse was happy. The owner was not happy. In fact, I said, this is kind of neat. You know, you can go riding, you know, got a place to put your cell phone, your wallet, you know, while your owner didn't think that was too funny. So we said, okay, so let's see if we can rebuild this. And so on the inside, we put in layers of SIS to undermine the mucosa, did the same thing on the outside. And now remember, this was 2 years duration. So in 6 weeks, it looks like that, and it's sort of, but a lot is filled in. Connective tissue beneath, now even on the inside, you couldn't see the hole from the inside, so we did another treatment, and at 12 weeks it looked like that, and at 18 weeks it looks like that. Her name was Classy, this horse. And we did an x-ray, actually. So on the inside's a normal mucosa, on the outside's relatively normal skin, the pigmentation's a little bit off, but it's much not. And on the inside there was a mixture of different types of connective tissue, fibrous connective tissue, bone, cartilage, whatever. So if we think about this from a science standpoint, the interesting thing is we basically filled this huge void with extracellular matrix, and the body did the rest. It formed skin on the outside, mucosa on the inside, and connective tissues, not a normal sinus, but connective tissues in between. So the body understood the microenvironment that dictated the differentiation of cells in the different locations, you know, really just a couple centimeters apart. Okay. This is something that I think you're going to hear a little bit more about from Dr. Ratner. We're working on esophageal applications too. This is a dog, and here's a trachea, and here's a 5-centimeter gap in the cervical esophagus. And we've taken a tubularized form of extracellular matrix, and if you take just half of the circumference of the esophagus and you put a piece of extracellular matrix in it does beautifully. Heals with almost no scarring whatsoever. As soon as you try to do the whole circumference, though, you end up with this, which is a stricture that has typical inflammation and scarring every single time. And it occurs between 17 and 22 days every single time. So we said, you know, that's not very good. We've got to get over that. So we then started mixing extracellular matrix with the adjacent skeletal muscle that sits normally on the outside of this piece of esophagus. And when we do that, the entire circumference ends up looking like this. We've got a nice mucosa, we've got organized muscle, and histologically it looks like this. The epithelium does not have the normal reedy pegs that help hold it in as well as it should. And this is the normal esophagus over here. Here's what used to be a piece of SIS, but you can see organized muscle has grown through here. We don't have any adnexal structures, and we've got an epithelial covering. And more importantly, there's normal motility, and there's not stricture. So this is an example where scaffold alone does not work. There are certain locations in the body where the interplay between the epithelial cells, the underlying mesenchymal cells, and appropriate matrix is important to get an appropriate, decent type of regeneration. So we're trying to we've taken this a step further, because right now the treatment for esophageal cancer is to remove your esophagus. You can't, even if you just have a little location of cancer, they don't go in and just remove the little location, because what happens if the esophagus scars? And if you get scarring in the esophagus, you can't swallow. And so what they take it out from the stomach up to the throat. It's called a gastric pull-up procedure. And it's not very pretty, and the quality of life is awful. So if you look over here, I don't know if you can see this very well, but this is a tube, that's the esophagus. They take out this part of the stomach and then tubularize what's left. Let's see if I can get this to work. Yeah, it goes like this. Okay, so they bring the stomach up through the chest, and it's attached there. Now, think of the problems that can occur. This is a surgical procedure where 50% of the patients end up with problems. If you think of any other surgical procedure in which 1 out of every 2 of your patients had infection or leakage with a mortality rate of around 6%, they would, say, you just can't do surgery. You don't know what you're doing. But that is the gold standard for this particular procedure, and it is the procedure of choice for this type of cancer. So we've got to come up with a solution for this. So what we've done, we've taken a conservative approach. We believe now we can reconstruct portions of the esophagus. But just like in the orthopedic world, if we took this scaffold to the types of surgeons, the cardiothoracic surgeons that do this and say, trust me, you can take out a piece of esophagus and put this in, they'll say, just that isn't going to work. No way. So what we're doing is saying, okay, go ahead and do your regular procedure. Take out the esophagus, pull this up, and let's apply a scaffold at that anastomosis. And let's see if we can at least reduce your 50% morbidity rate. And oh, by the way, when you look in there, you're going to see restoration of normal mucosa and esophagus. Then they're going to say, well, let's see if we can go a little bit further. Let's go down and start taking out pieces of tumor rather than the entire esophagus. And then eventually they can remove it. It's basically a strategy that we're employing that will probably take 6 to 8 years to commercialize. But the idea is that we make a funnel-shaped device out of the ECM and then slide it into just that area where the stomach is brought up and anastomosed to the esophagus. And I've got a picture. I just tried this before I went up there. It's not going to work. This is an endoscope that shows basically the animal procedures that led to this and shows a restoration of a normal mucosa. Right, you can't even see where the anastomosis is. And so let's see. This is what it looks like in the animal experiment. Esophagus coming down, stomach in here, and what we've done is placed on the inside of that a piece of extracellular matrix scaffold. And what you can see now is where the esophagus now enters into the stomach, it's covered by essentially normal mucosa. This used to be nothing other than porcine-derived extracellular matrix at this location. Now, if this same result can be obtained in humans, and we're starting 2 human clinical trials in the next 2 quarters, one in Buenos Aires and one in Pittsburgh, then I think we'll be able to convince surgeons that we've got something that can at least modify the default mechanism of healing in the esophageal tissue. So we've learned from previous experience, we're taking a very conservative approach to translating this into the clinical community. Here's what this looks like when you open it up. Esophagus coming down, everything from about here down to about here. It was nothing other than ECM before, and now we've got a normal transition in here. And histologically, it looks like this. On this side over here, here's the gastric mucosa, here's the esophageal mucosa, here's the anastomosis right in here, and we've got a normal submucosa, and if I showed you really close, you'd see islands of the muscle layer here is continuous. This is what we call the clinical control, where we simply made an incision, sewed the 2 ends together, and didn't do anything with the mucosa, and every time we get this deep invagination in here, we lose the muscle layer, and here's a suture down here. We've got a chronic inflammatory reaction that persists there, and this is what ends up being scarring. Simply if you take an incision in the esophagus and pull it together with the stomach, which is why there's always stricture that occurs. So somehow by just placing the ECM in there, we change this to this. That's happened in 2 animal models. Let's hope it happens in people. So our clinical translation strategy, basically our commercialization strategy on this, is a little different than we did with SIS. What we're going to do is take a high-risk anastomotic procedure, allow them to continue to do that, advance to the treatment of Barrett's esophagus, which is a pre-neoplastic condition, go to things like esophageal cancer and caustic esophagitis, and we're thinking it's going to take 6 to 8 years, but what we're doing now, and we have not yet gotten our industry partner. So our industry partner is going to come within the next couple of years, because we wanted, in this case, to get human clinical data prior to getting the industry partner. And obviously the value of that technology changes significantly with that type of a strategy. Let me show you one more. I don't want to run too long into, I know we started a little late, but I don't want to get too long into our break time here. This was another interesting one. This is a dolphin. I got a call from a veterinarian in Hawaii, this is the fin of the dolphin here, and there's this huge gap here. And this dolphin, they've tried to fix this 3 times, and the problem is this is not being pulled back, it's just missing. And a dolphin's fin is important not just for navigation, but it regulates temperature, does a lot of other things. And they were worried that if he bumps this one more time, he's basically going to rip it off. So they said, is there anything you can do? So a couple of graduate students in my lab went to work, and they custom designed the urinary bladder matrix scaffold for this. And they flew one of my techs down to Hawaii, which wasn't too bad of a problem. And this is what the looks like. Here's the fin coming up here. Here's the dolphin. And there's this gap with this exudate in here to start with. And whoops, yeah, there we go. So it looks like that. You can see that you can actually slide material back into there. What we did was then cleaned it up, filled it with a particulate form of the ECM, and then made a funnel-shaped device that we were able to put around it. And they made a special vest for him. He'd run around like that to keep it in place because you can't keep him from swimming. I've gotten more calls on this particular procedure than any human we've ever treated with SIS. I still get calls. This was done 2 years ago. I get calls about every week saying, how's LECO doing? And what did you do? And we've got, a parakeet over here. We've got, so I'm doing more in veterinary medicine now than I am in human medicine. So this is what it looked like 12 days post-op. Already we got a proliferation of tissue in here because obviously we had to bring him out and sedate him each time. And then you can see this is imaging it, you can see a very aggressive angiogenic response in this new tissue. And this is what he looked like this was about 10 months later. So even though it's not perfect, we basically have healed this totally back down, and this happened about 2 years ago. So he's doing, now this was challenging because think of the environment. What does healing like occur in 3% saline? And in motion, constantly, this thing's, being exposed to, and this was a pretty aggressive male dolphin. So that was LECO, so he's happy. Another quick one. This is a device that SIS is being evaluated for as a venous valve. It's spread along a metal spring here, and we've implanted it into the jugular vein of sheep for chronic experiments, and interestingly, if that stratum compactum surface faces out, it's relatively non-thrombogenic. So we've got a material here that started out as pig intestine and now looks an awful lot like a valve. You can see the metal still here along the edges, and this is what it looks like histologically. Here's the wall of the vein. Here's what used to be a piece of SIS, and it's covered by endothelium. So this is currently in clinical trials in South America, and it's delivered obviously by a catheter. There's the endothelial cells that look like it. Another quick one. This is the larynx of a dog, and what we've done is we did a hemilaryngectomy. We took out half of the larynx, everything from here down along the middle, out to here was completely gone and replaced by a piece of extracellular matrix. So the point of this is that the same matrix is being used in a lot of different locations, but the end remodeling result is entirely different. This didn't turn into bladder, it didn't turn into a dolphin's fin, and it didn't turn into a piece of Achilles tendon. In this location, this turned into a larynx. And we've got, we made a single layer sheet in here, it turned into a vocal fold, and histologically, it's even more interesting because here's the muscle that's reformed. This is the cartilage plate that used to be a piece of ECM. It's got a layered area of skeletal muscle in here. Here's the vocal fold coming around. Here's a piece of cartilage that doesn't belong there. For some reason, we got an island of cartilage forming in the tip of that vocal fold, but there's also glandular structures in here. So we got a very interesting self-assembly of different connective tissues simply by providing an environment for remodeling. If you did this just with collagen, you end up with scar tissue. So what is it that's unique about the structure and composition of an ECM that can form these sorts of different tissues? And one other one, we got a clinical trial going for a meniscal replacement, which this is a preclinical study showing that if you take the middle third of a meniscus out, it can end up looking like that in a very short period of time. And so this is in a phase 1 clinical trial now at 4 different centers in the United States. Okay, let me show you one more. This is a pretty interesting. Huge holes in the sides of animals. This is like 8 centimeter hole, full thickness defect, replaced with a multi-laminate sheet of an ECM, and in a matter of 5 months, it ends up looking like this. Now, we didn't put a skin graft over that. The skin grew back over that. So this is being used for trauma in the United States. And I see our leader up here. It's time to quit. The last thing I was going to show you was a follow-up on the mechanism, which had to do with the bioactive peptides that are a result of the degradation of the ECM. So I'll stop, but what I wanted to do was sort of give you some ideas of what we think it takes to commercialize or translate a university-based discovery to patient care, and where we are in trying to understand the use of this scaffold for a variety of clinical applications.
Thank you. Let's take a few minutes for questions, and then we'll move into a break. Are there any questions? Yes.
Actually, I would like to ask about the status of credibility of clinical trials performed outside of the U.S. or the regulatory body in the U.S.
So we actually are using that strategy in some. We've got clinical trials that are occurring in South America. The FDA, they typically take the stance of saying, I don't care what you've done outside of the United States. You still have to do everything that you've done in the United States that way. However, we learn a lot by being able to do so we do very small clinical trials outside of the United States, 10, 20 patient pilot type studies. And we basically take the same approach as we would in the United States. But I think maybe one of the things that helps with is we can get a lot of data, and I'm not quite sure what the change was in the clinical study with the diabetic foot ulcers and dermograft, but maybe if that sort of thing would have happened, or maybe not, outside, in the first 20 patients, you know what you need to change before you come back and spend a lot of money on a clinical trial in the States. So that's our approach.
Are there other questions? Barbara.
Yes, please.
You got a microphone here.
Thank you for the wonderful presentation. I have a question concerning, as I understood, that all with SIS coming from the porcine animal, that's right?
That's right.
What about the immune response you've applied in many species? So what about the immune response in this case?
Yeah, I put together an immunology advisory board, I called it, in 1995, and we met once a year. They would come and say, here's what you need to do. We work for a year, come back and tell them. We did that for 6 years because we're not immunologists. And I rationally said, there has to be an immune response, even though we eliminated all the cells. And sure enough, every patient and every animal that receives a xenogenic extracellular matrix mounts an immune response. We can measure antibodies, and then we try to say what type of immune response is it. And I'm not sure if you're familiar with this. There's a paradigm in immunology called a T helper 1 type response versus a T helper 2 type response. One has to do with typical rejection, and another has to do with what's called accommodation. Every single time that you take the matrix and don't have any cells in it, you get the Th2 accommodation response. So there's a profile of cytokines that are produced that accommodate the graft. It's sort of like the response that a woman has to her fetus. It's a foreign antigen, and I used to say that instead of rejecting this fetus immediately, it takes about 16 years before you reject it out of the house to go to college. But it basically changes. The point is there is an immune response. The body recognizes it as foreign. I think one of the keys, though, is it rapidly goes away.
Okay, so
What is the time course?
You're talking about the fibrous normal response action, about the fibrous capsule, like encapsulating the graft, or what?
No, there is no fibrous. They
fibrous
integrate.
capsule even surrounding this the matrix?
Never. We never get a fibrous.
Never get that capsule around.
That's what's key. The cells recognize it, they attach to the surface, they grow into it, they degrade it, and it goes away.
So it's fully adapted by the surrounding tissue?
Yes, it's degraded. If we cross-link it chemically, then we get a fibrous capsule, because now the body cannot break it down. In response to the timing of the immune response, it's immediate. Within a week, we see the initial IgM response, and then we end up with peaking at around 8 weeks. And by 6 months, we can no longer find it. And if we reimplant, we don't get a sensitization and a Th1 response. We actually boost the Th2 response. We've got about 6 publications that are characterizing this. So it's pretty well documented in the literature.
We need to take one more question, and then we're going to go to a break. Barbara, you're next. You can talk to him during the break.
Yes, because I have a second question maybe later.
You can ask during the break. Go ahead, Barbara. Just shout. You're good at it.
Thank you for that. My question has got to do with more practical
the point is, you're talking about what do I think industry needs to say this is a good bet?
Yeah.
You know, as opposed to as if we got lucky one time. The approach we take now is that we look for statistical significance. We put n values of 6 in each group, and we try to get at least 6-month, if not 12-month data, because they all want long, and we do it in 2 species. If we take that sort of data to a potential industrial sponsor, they really have a hard time arguing with it. Now, maybe you don't need that much, but I've become more conservative rather than more risk-taking as I've gotten older. And it's, so I look for 6 to 12-month data in 2 different species in the application.
I've noticed you're becoming more conservative.
Yes, Abby, thank you. Debbie, really, we won't comment about that.
Okay. Let's take a break and be back here at 11:15. I assume over in the restaurant there's coffee and so forth.